Hello everyone, I'm Dr. Arpita De. The topic for today's discussion is citric acid cycle, or otherwise known as tricarboxylic acid cycle or Krebs cycle. This is one of the most important lesson in our physiology unit. Citric acid cycle, as we all know, is the second step of aerobic respiration, the first being glycolysis, where glucose is broken down into two pyruvate molecules. I would like to suggest to people to first go through my glycolysis lecture. In case if you have missed it out, I'll share the link of glycolysis lecture at the end of the video. Kindly go through that before coming to this. Okay, so let's start. So citric acid cycle is also known as the TCA cycle or tricarboxylic acid cycle or Krebs cycle. Okay, now see, TCA cycle is a metabolic pathway that has a series of chemical reactions which leads to the release of stored energy through oxidation of acetyl coenzyme A. Okay, this you will have to remember. Now see, you must also know where is it taking place. So this takes place in the matrix of and the number of steps involved are 8. Here we are going to discuss each of these 8 steps in detail, right? Now see, citric acid cycle is the second step of aerobic respiration in all living organisms, right? Glycolysis being the first. Glycolysis is the first step of aerobic respiration where glucose is broken down to two pyruvate molecules. You all know this. As I've already said, if you are not aware of this fact, please go through the lecture on glycolysis. I'll share the link in the comment box or in the description box, right? See, oxidative decarboxylation of pyruvate yields acetyl-CoA, which takes place in the first reaction of TCA cycle, right? So let's start with the very first step here. See, oxaloacetate is a 4-carbon compound. It's a 4C compound, right? Acetyl coenzyme A is what? It's a 2C compound or 2-carbon compound. The acetyl coenzyme A molecule combines with oxaloacetate to give citryl-CoA, right? Citryl-CoA will yield citrate or citrate. This reaction is a condensation reaction which is mediated by the enzyme citrate synthase. So what type of reaction is this? This is a condensation reaction. Condensation reaction, right? And is mediated by the enzyme or A. I've written letter codes for enzymes for all the steps here. The enzyme is citrate synthase. Right? Coming to step number 2. Step number 2 is a dehydration reaction. Okay? What type of reaction? See, the previous one was a condensation reaction. This one is a dehydration reaction, right? Why is it a dehydration reaction? You can understand. A molecule of water goes out. Okay, so citrate forms cis aconitate after this dehydration phenomenon. Then what is happening? See, another step of hydration occurs here. Addition of water. Okay, so what can we call this? Is hydration. We can say hydration is happening because water comes into play, and cis aconitate is converted to isocitrate. Fine, this is an isomer of citrate, you can understand. See, both these are reversible reactions, and this reaction is mediated by enzyme B, which is aconitase. Okay, fine, this is aconitase. Now I don't know whether you are aware of the term suicidal inhibition. Suicidal inhibition is a poisoning phenomenon. From the word suicide, you can understand what happens. See, the poison fluoroacetate is toxic because fluoroacetyl-CoA condenses with oxaloacetate to form fluorocitrate, which inhibits aconitase. That means this enzyme gets inhibited, right? It inhibits aconitase, causing citrate to accumulate. Okay, if this is inhibited, that means what? Isocitrate will not be formed and citrate accumulation occurred. This phenomenon is called suicidal inhibition. Fine. Next we'll move on to the third step. Third step is an oxidative decarboxylation reaction step. What happens here? See, oxidative decarboxylation takes place here. What is happening? See, NAD plus gets converted to NADH plus H plus, while isocitrate is getting converted to oxalosuccinate. Okay, so we can write dehydrogenation here, right? Then you can see this oxalosuccinate. Okay, this oxalosuccinate is forming alpha ketoglutarate, right? This oxalosuccinate is giving us alpha ketoglutarate here, which is mediated by an enzyme called isocitrate dehydrogenase. Fine, isocitrate dehydrogenase. Isocitrate dehydrogenase, right? Coming to the next step, step number 4. Here you see alpha ketoglutarate is getting converted to succinyl-CoA along with the production of carbon dioxide and NADH compounds, right? See, here the enzyme which mediates this reaction is alpha ketoglutarate dehydrogenase. Keep in mind that this alpha ketoglutarate dehydrogenase complex is inhibited by succinyl-CoA and NADH. Okay, these are some very important small little points which are often overlooked, but they are really very, very significant and you must know these. Another important point about this step number 4 that you should know is that arsenite is an inhibitor of step 4. So if I just write here, Step 4 is arsenite, right? What does it do? See, this inhibits arsenite, inhibits this step, and as a result what happens, see, it results into accumulation of alpha ketoglutarate. Correct? Because if this step gets obstructed or inhibited, what will happen? This alpha ketoglutarate will not get converted to succinyl CoA. Now step number 5. The succinyl CoA formed in the previous step now gets converted to succinate. Okay, and you get coenzyme A and GTP. Now see, this GTP formed here is used for decarboxylation of oxalate to PEP in gluconeogenesis. You will have to remember this. The enzyme here is E, which is succinyl CoA synthetase. Okay, I'm writing the enzyme. Succinyl CoA synthetase. This is the step where we can see substrate level phosphorylation. You can see this here in this step. We can see phosphorylation occurring at substrate level. GDP is getting converted to GTP with inorganic phosphate. So we can call this step as, writing it with a different color so that you remember it. It is called substrate level phosphorylation, right? So step number 5 is your substrate level phosphorylation step. Next, coming to step number 6. In step number 6, what happens? This succinate formed here is now reacting with EFAD. What is this EFAD? EFAD is enzyme containing FAD. EFAD is enzyme containing FAD, and this reaction is a reversible reaction which is mediated by enzyme F. F is for succinate dehydrogenase, right? F is for succinate dehydrogenase, correct? This succinate dehydrogenase is also inhibited by oxaloacetate. So oxaloacetate has the potential to inhibit this particular enzyme, that is succinate dehydrogenase, correct? So what is formed here? Fumarate is formed. In step number 6 we get fumarate. This fumarate in step number 7 gets converted to malate. Fine. The enzyme here is fumarase. Okay, the enzyme here is fumarase. See here, fumarase catalyze addition of water. You can see a molecule of water comes in. Remember, fumarase catalyze addition of water. Please remember this. Then coming to the final step of the reaction, where this malate from the 7th step is finally forming oxaloacetate. Okay, Oxaloacetate is formed here once again. Now you see, why do we call it a cycle? Why is it a cycle? See, we have started this TCA cycle with oxaloacetate, that 4-carbon compound. Once again at the end of the cycle, we are coming back. At the last step, we are coming back to the substance with which we started. That is why it is called a cycle. Here what do you see? See, malate plus NAD gives oxaloacetate NADH plus H plus in a reversible reaction mediated by enzyme H, which is malate dehydrogenase. Malate dehydrogenase. Correct?